Hi folks, welcome to AB Biopharma with Dr. D. This is part 4 of the series of how structure determines function, a tale made of biomolecules. And I would like to remind you that these videos do not instruct you on the basic structure and the functions of biological molecules. This is simply to illustrate how their structure and how changes in their structure can actually change their properties and determine their function. So this is not an introduction to function of lipids. So let's talk about lipids. Okay, the first class of lipids we're going to be talking about are triglycerides. They're the most abundant animal fat. They're formed by the joining of 3 fatty acids to a glycerol molecule. Glycerol, it ends in OL. That means it's an alcohol. And how do you know that it's an alcohol? It has lots of hydroxyl groups. So the glycerol is used as a backbone, and a dehydration reaction occurs, removing a hydrogen ion from each of the hydroxyl groups of glycerol, and the OH group from the carboxyl group of the amino acid, and it forms an ester bond. And you attach 3 of these, 3 fatty acids, to a single molecule of glycerol, because the glycerol has 3 hydroxyl groups, and therefore we call this a triglyceride. 3 molecules of water will be released in the process, and this is again a dehydration synthesis or a condensation reaction, because water is released. Okay, dehydration synthesis or condensation reaction. There are 2 types of fatty acids: saturated fatty acids. They're called fatty acids because of the carboxyl group, and the carboxyl group is a negatively charged acidic group, if you remember. Saturated fatty acids have no double bonds. They're saturated with hydrogen ions. So a fatty acid contains mostly carbons and hydrogens, and the only oxygen can be seen in the carboxyl group. So every carbon within a fatty acid has exactly 2 hydrogen attached to it, except the ones at the end, which have 3. So the carbons are saturated with hydrogens. Now, if you were to introduce a double bond right here, what you notice is, because of the valence of carbon of 4, there's now only 1 hydrogen attached to each of these 2 carbons, as opposed to 2. So this fatty acid is not saturated with hydrogens. You could add some more hydrogens. If you were to break that double bond between the carbons, you could add 2 more hydrogens to this fatty acid. For that reason, this fatty acid is called unsaturated fatty acid. Saturated fatty acids have long hydrocarbon chains, again, connected by a single bond. Unsaturated fatty acids have 1 or more, they could have more double bonds, and each double bond may be either in a cis or trans configuration. Why am I telling you this? Because you have heard of trans fats. This is where the term comes from, trans fats. So cis acid looks like this. The hydrogens are positioned on the same side of the double bonds. So this acid will bend. It will bend because of the double bond. Now, if the hydrogens are positioned on opposite ends, which is the lowest picture, which is a trans acid, there will be no bend in this acid, and that's important. The trans configuration, the hydrogens are on the opposite side, and the bend will not exist. So if you look at a trans acid and a saturated fatty acids, they kind of look the same, one straight line. A cis unsaturated fatty acid has a bend, has a kink. And here are some other models, representation of a trans and a cis unsaturated fatty acid. This is a saturated fatty acid. This is a cis unsaturated fatty acids, and this is a trans unsaturated fatty acids. Why am I talking about this? Well, it is important to understand that they're well-defined detrimental effects to health. So it's really bad for your health if you consume trans fats. There's a strong association between consumption of trans fats and heart disease. Not all fats are bad. As a matter of fact, fats are really good for you. However, trans fats in large amounts are really not good for your health. There is no physiological need whatsoever for us to consume trans fats. Trans fats occur in nature only in a very, very small degree, but it's recommended to avoid manufactured trans fats. So how do we get trans fats in the food? In nature, most fats are either saturated fatty acids or cis unsaturated fatty acids. If you're cis unsaturated fatty acids, then you're an oil, you're liquid fat. If you are a saturated fatty acids, then you're something like butter or lard, you're a solid fat. So people at some point thought that solid fats are bad, butter is bad, lard is bad. So plant oils are really good for you. They're really good, but plant oils are like this and they are liquid at room temperature. So how do we make spreadable oil? Well, we turn it into margarine. We partially hydrogenate this fatty acid. So we artificially add some hydrogens and destroy some double bonds. And in the process, what happens is we end up producing a trans fatty acid. So margarine, I can't believe it's butter, are very rich in trans fatty acids. That is an artificial process that is designed to convert liquid oils, plant-based oils, into something solid and spreadable. Now, manufacturers are now required to list the grams of trans fats on a label, but manufacturers actually can get away with not telling you that their foods contain trans fats. So they can label 0 grams on the nutrition panel, even if there is 0.5 grams of fat. So to figure out if a product has trans fats, look on the label, and the label should say that it contains partially hydrogenated oil on the ingredients list. So if the ingredients list says partially hydrogenated oil, then your food contains trans fats and you want to avoid eating too much of it. Okay. Now, the other important class of lipids are the phospholipids. And here, R stands for anything goes. What does the phospholipid look like? The phospholipid contains, again, glycerol, except the glycerol has only 2 fatty acids attached to it. And the last hydroxyl group, instead of having a fatty acids, has a phosphate group. A phosphate group is negatively charged, if you remember, and therefore is hydrophilic. In addition to the phosphate group, there are some other groups that are also polar or charged. So R basically varies between the different type of phospholipids. So different type of phospholipids have different R groups, but all phospholipids have a phosphate group. So the head of a phospholipid contains the phosphate group and some other charged and polar groups, and the portions of glycerol, and it's highly hydrophilic. The tails are hydrocarbons, saturated or unsaturated, and they're hydrophobic. And this is a familiar picture of the phospholipid bilayer of the cell membrane, where the hydrophilic heads are facing either the outside of the cell, which is water-based, or the inside of the cell, which is water-based, and the hydrophobic tails are buried inside of the bilayer. Here's an example of one of the famous phospholipids called phosphatidylcholine. You don't have to remember the name, it's just I wanted to show you a picture and a formula of one such phospholipid. The 2 fatty acids, the 2 glycerol, the glycerol backbone. The 2 fatty acids are attached to the 2 of the hydroxyl groups. This is the phosphate group, and to it we have something attached with 2 methyl groups and a nitrogen and a methyl group, and it's not really important what exactly this is. This is called choline. The important thing is that this whole part here in gray is hydrophilic. Why? Because it contains either charged groups, negative charge here, positive charge here, or polar covalent bonds, okay? Whereas the rest of the molecule are the hydrophobic tails, okay? Another important lipid is cholesterol. Cholesterol is in the group of sterols. Sterols form ring structures. Cholesterol helps keep cell membranes fluid and flexible, and we're going to talk a lot more about cholesterol when we talk about the structure of cellular membranes. From cholesterol, you can also make sex hormones, steroid hormones, such as cortisol, aldosterone, testosterone, estrogen, progesterone, are all made as modification of cholesterol. Cholesterol can also be used to make vitamin D. Believe it or not, vitamin D is a lipid. It's a steroid. It's a sterol, not steroid, because if I say steroid, you're gonna think steroid drugs. So UV light, with the help of UV light, we can actually turn cholesterol into vitamin D. This is how we make vitamin D in our bodies. All right, now remember, fats and lipids are mostly hydrophobic. They do not like water. And this is the end of our video. Till next time.